Hey everyone, welcome back. I'm just changing my color so I have the color I like. All right, so now we're going to talk about transcription. Specifically, what is transcription? So we're taking the DNA and we're converting it to RNA. That's what transcription is. And I always think of this as taking language that maybe you can't read and getting it converted into a language that you can read. So if you can't read German, but you get directions to build a crib in German, you're not going to be able to very effectively build that crib, right? You have all the parts to make a baby crib, but you can't build it because you don't understand the language. So if you get it converted to English, which you can read better, now you can follow the directions, right? So DNA is a language our cells do not read. They can't read. It could, I can't tell you why that we can't read DNA. All I know is that our cells don't use DNA to produce the proteins directly. They have to convert it to RNA. So RNA is the language that our cells use. This is a language that our cells can read. So we take DNA, we convert it to an RNA, and then that RNA can go out and actually be used to produce a protein. And that is transcription, okay? Taking the DNA and converting it to that language that we can read or we understand. So we have to have certain enzymes. In replication, we used an enzyme called DNA polymerase that allowed us to add DNA nucleotides. In transcription, we're going to use RNA polymerase. RNA polymerase binds to a region known as the promoter site. Once it binds, it then can move along the DNA and it can allow for RNA nucleotides that is an E, to come in and bind. So the RNA nucleotides that are complementary to the template come in and they form that RNA. This RNA polymerase reads the DNA all the way to a region called the terminator sequence. So once it hits that terminator sequence, the RNA polymerase comes off and the DNA can wind back up. So this is what it looks like. In this case, this bottom strand here is our template strand. So this top part here is called our coding strand. That's the strand that we're actually making a copy of, okay? And then if you look at the messenger RNA, as it comes in, anywhere that we have a T up here, we're going to put a U in down here. And this is just going to keep going all the way along. So what we'll produce is an identical copy of this region of the DNA in an RNA format, okay? And so let's look at what that looks like. And so they kind of have, right, starting here, you have the C, you have the A. Anywhere there's a T, you have a U. C, C, A, A. There's another U, and then another U's coming in. So you can kind of see that it looks the same. So let's look on the next slide at what that would look like. Oh, I'm lying. So this it would have been great if they then showed a bigger picture of this, but they didn't on here. So I have my own example. So this is something I use in my microbiology class. I made this up to show transcription in organisms. And so this is showing, it says prokaryotic, but it doesn't matter if this is prokaryote or eukaryote. Transcription is still going to be the same. It's just where is it taking place is going to be different. So again, we have a coding strand. The coding strand is what codes for the protein. So down here is where that protein is actually coded for. We're going to use the template strand we use to make a copy of the coding strand. Okay, so we use the template strand, and we allow for DNA polymerase, or RNA, I'm sorry, RNA polymerase comes in, it's going to bind. It binds only to a 3' end, just like in replication, we still can only bind to that 3' end. It binds that 3' end of the template strand and allows for the proper nucleotides to come in. So this is RNA polymerase. I'll put that right here. RNA polymerase allows for the proper RNA nucleotides to come in. So let's look at those proper RNA nucleotides. So if our DNA has an A in it, which you see right here, right, then you're going to have in RNA, that's not correct because that wouldn't show what I want. In RNA, you're going to have a complement called uracil. If you have a thymine in the DNA, you're going to have adenine. If you have a guanine in DNA, you're going to have a cytosine. And if you have a cytosine in DNA, then you have a guanine in RNA. So the RNA nucleotides are complementary. The major differences in DNA, we have thymine. In RNA, we have uracil. So let's look at what that looks like. And so I didn't actually put in all the stuff, but here we have the RNA polymerase is going to be moving along in this direction. And the nucleotides, RNA nucleotides from the nucleotide pool will come in. Anywhere we have an A on this strand, we're going to put a U. Anywhere we have a C, we put a G. Anywhere we have a T, we put an A. And anywhere we have a G, we put a C. So what you can now look at, this is the mRNA, right? And I want you to look at this and compare it to this, because this is our coding strand. So the difference is anywhere you see a T in DNA, you'll have a U in RNA. So here we have a U and a T, a U and a T, a U and a T. G, 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 A, U again, T, G, G. G, 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 C, 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 C. So do you notice that's exactly the same? They're identical. This messenger RNA is identical to this coding strand of DNA, except for where you have thymines in the DNA, you have uracils in the RNA. So once we make that messenger RNA, if we are in a eukaryote, this all occurred in the cytoplasm, or I'm sorry, this all occurred in the nucleus. And before we can leave that nucleus, we have to process that messenger RNA. So this occurs only in eukaryotic cells. In prokaryotes, we don't have to process our messenger RNA. It's already ready to be translated. But in eukaryotes, we have to process it. So we call it immature or pre-messenger RNA. And so the things that we have to do, we are going to take out in our DNA. There's a lot of material that doesn't code for proteins. These sections are known as introns. An intron is a non-coding section. So I think of introns as, if you're watching a movie, think of, probably everybody has watched a Lifetime movie in their lifetime. Kind of a joke there. Okay. Anyways, so when you're watching a Lifetime movie and you're getting really into it, something exciting is going to happen, and then they go to a commercial. That intron would be the commercial. It's just it doesn't code for the movie that you're watching. It doesn't have anything to do with the movie. It's just there. Okay, a lot of these introns are regulatory regions, but they aren't part of that protein, so we have to get rid of them. So we have to get rid of the introns, and then we have to put together, splice together all of the exons. Exons are the expressed portions of the DNA. So think about taking a movie that has commercials and taking out all of those commercials from the movie, so you have a complete movie where there's no breaks. That's what processing does, okay? Along with processing to get rid of those introns, we also add a guanine cap. So what's a guanine cap look like? It's a bunch of guanine nucleotides at the beginning of that messenger RNA. We add a poly-A tail. That's a bunch of adenine that are bonded to the end. And once we do this, this is now considered mature messenger RNA, and we can send it out to the ribosomes to be translated. So this is a very small picture, but you can still see what's going on. Here you have those light regions. Those are the introns. We're going to get rid of all those light regions. So first we copy it, and we now have the messenger RNA. Messenger RNA still has those introns and exons, but we only care about the exons. So we use a protein enzyme. Actually, this is a ribosome enzyme, or an RNA enzyme called a spliceosome. And spliceosomes take out those introns and splice together the exons. So now we have the exons, we have that guanine cap, and the poly-A tail. And I think I like this picture. It's big, and so you can see it even better. So here we have the DNA. These are the introns that don't code for the protein right here. We add a guanine cap. We add a poly-A tail, and we get rid of those introns and splice together the exons. And so this is the mature messenger RNA that can leave the nucleus and go into the cytoplasm. This only occurs in eukaryotes. Prokaryotes, we don't have to worry about any of that. And so I'm going to stop here, and we'll get into translation in our next video. Bye.